Hello, welcome. The practical work today is about plant cell wall. The objectives of this practical work. At first, we have to make the difference between plant cell and animal cell. We have to know the organization and the composition of the primary wall and the secondary wall. And finally, the highlight the intercellular communications of the plant cell. The eukaryote plant cell has the same organelles as the animal cell. However, eukaryote plant cells have some characteristics which are the presence of pictocellulose wall, geometric shape, presence of chloroplasts, presence of a large vacuole, and absence of centrosome. The cell wall is a rigid covering that surrounds the cell. It makes a separation between the interior contents and the exterior environment. It provides shape, support, and protection to the cell. The plant cell wall is organized into three layers. We can see them on the TEM micrograph of ultrastructure. These layers are the middle lamella, the primary cell wall, and the secondary cell wall. The first one, the middle lamella, which is shared by adjacent cells, it's made up of pectin. The second one, the primary cell wall, it's made up of cellulose microfibril, hemicellulose, pectin, and water. This layer is flexible to allow for cell growth. The third one is the secondary cell wall. This layer is thicker and more rigid. It's richer in cellulose microfibrils, poorer in hemicellulose and water. It lacks pectin and glycoprotein. This layer provides additional support for the cell and helps to protect it from environment stress. In this micrograph, we can see that young cells are made up only of the primary wall, which is essentially composed of water in order to give flexibility to the cell to grow. The observation of the primary wall using SEM shows an anarchic arrangement of the cellulose microfibrils. The secondary wall is made up of three layers, which are S1, S2, S3, where the cellulose microfibrils are arranged parallel to each other in each layer, and their orientation is different from one layer to the next. These layers are very low in water, which gives them great rigidity. In this micrograph, we can see the cellulose microfibrils are parallel to each other. We can see also that their orientation is different from a layer to the next. Here we can see clearly the difference between primary wall and secondary wall on SEM micrograph. In the first one, we can see that the cellulose microfibrils are anarchic, but in the second one, they are parallel to each other. The intracellular airspace is formed when the middle lamella breaks down by an enzyme called pectinase. This leaves a void between three cells. The intracellular airspace helps to circulate water and gases between the cells. Now we are going to speak about intercellular communication. After the formation of the plasma membrane, the primary and secondary wall, there remain in places spaces that will constitute the plasmodesmata crossed by the smooth endoplasmic reticulum. In this micrograph, we can see a longitudinal section and a cross section of the plasmodesmata in a firm gametophyte observed under the TEM. Punctuations or pits. There are structures that are visible under a light microscope. There are two types of pits. Simple pits, thinning or interruption of the wall, the primary one or the secondary one, and maintenance of the middle lamella. The second type is called bordered pits. It's a continuity of the middle lamella and the primary wall. The primary wall forms a central thickening called a torus. The secondary wall is interrupted. Thank you for your attention. See you next video.